Welcome students. In today's class I am going to teach you about spleen. Spleen जैसे हिंदी में तिल्ली कहते हैं. As I have already told you, in anatomy, whenever we learn about any organ or structure, first thing is we should know the location of that organ, then the relations, shape, size, the position, exact position in the body of that particular organ. After that, we have to see the relations of that organ, then the macroscopic and microscopic function of that organ, and then the applied anatomy. So first of all, I will tell you the spleen that you can see very clearly. It is a wedge-shaped organ. Okay, it is wedge-shaped organ. And what is the position? So for telling the position of any abdominal organ, we have to know what are the regions in which abdomen is divided. It is divided into 9 regions. Right hypochondrium in the upper right part, left hypochondrium on the upper left part. In between the two, there is epigastrium. Below the right hypochondrium is the right lumbar region. Below the left hypochondrium is left lumbar region, and in between is the umbilical region. Below the right lumbar is the right iliac fossa, and below the left lumbar region is left lumbar iliac fossa, left iliac fossa, below left lumbar region. Okay, and in between hypogastrium. So this spleen, it is lying in partly in epigastrium and mostly in the left hypochondrium. So this is the organ which is present on the upper left side of abdomen. In the right hypochondrium, we will find liver. Liver is a large organ, so it is going to occupy more than right hypochondrium. It will also have its position in the lumbar region, right lumbar, and epigastrium also. So in between the two were stomach and pancreas, which you have already studied in previous classes. And now we come to the extreme left, and that is the spleen. So spleen, it is lying in the body in the left hypochondrium as well as a little bit of epigastrium. So these are the two regions occupied by the spleen. So as this wedge shape will tell you, the wedge shape is because the organs which are in relation to it, because all the organs which are coming into contact with the other organ are going to put a pressure, and because of that pressure effect, an impression is formed, and so the shape is obtained by the structure. So first of all, now the size, because we have confirmed where it is lying in right hypochondriac region as well as epigastrium. Now what is the shape, size, weight, and the direction in which it is present? Okay, so it's very simple and very interesting too. Just see the odd numbers: 1, 3, 5, 7, 9, and 11. So these are the numbers which you have to learn. It's so simple. So it is 1 inch thick. Okay, so thickness is 1 inch. If you convert into centimeter, 1 inch is 2.5 centimeter. You can always multiply by 2.5 and you will get the dimension in centimeters. So 1 inch is the thickness. Next odd number is 3. 3 inches is the breadth. So the breadth is 3 inches. So again, you can multiply by 2.5 and you will get the answer in centimeter. But it's easier to remember and more interesting also if we talk about the odd numbers. So 1 inch is thick, 3 inches broad, and next number is 5. 5 inches long, 7 ounce in weight. So its weight is 7 ounce. Now the odd numbers which are remaining are 9 and 11. So you will say, we have talked about the thickness, we have talked about the breadth, we have talked about the length. What is left? Yes, there is definitely the left. That is the position. Although we have learned that the regions it is occupying is epigastrium and left hypochondriac region, but still the exact position, the exact location. So it is lying between 9th and 11th rib. So that is the significance of 9 number and 11 number. So it is lying between 9 and 11 rib. That means the 9 rib will come somewhere here and 11th rib will come somewhere here. So what is the axis and the direction of spleen then? Since it is lying between 9th and 11th rib, axis is going to be along the 10th rib. So the axis of spleen is along the 10th rib and it is directed, see, obliquely downwards, backwards and outwards, because it is lying along the ribs. So this is about its dimensions. Now we will talk about the surfaces. It has got 2 surfaces. This you can see lot of impressions are there. So impressions are always created by organs. Organs are called viscera. This is the visceral surface. Next is the diaphragmatic surface. This one. You can see it is convex, no impressions. So this is diaphragmatic surface and this is visceral surface. First I will talk about the diaphragmatic surface. This surface, diaphragmatic surface, is related to diaphragm. First of all, above the diaphragm, pleura, then base of left lung, and of course on the outer side the ribs, 9th, 10th and 11th ribs. So these are the relations of diaphragmatic surface. Clear? Now I talk about the visceral surface. See, this impression you can see, this is gastric impression, means this is related to stomach. Stomach has put this indentation over here. Then in front again you can see. So this is colic impression. That means the colon, because this is the left, extreme left side. So we are going to have, this is actually the impression produced by the transverse colon and descending colon. The junction of these 2 colons is called splenic flexure. So this is the impression of that splenic flexure. That is why it is called colic impression. Then left is one more impression, and this is present on the inferior surface, inferiorly. So that is produced by left kidney. So it is renal impression. And in last class when we were talking about pancreas, I have told you that tail of pancreas is going to put an impression on the hilum of spleen. So tail of pancreas will have impression here at the hilum and approaching a little bit on the renal impression. So these are the impressions on the visceral surface of spleen. Now, so you know what is the position of spleen. Now you know the dimensions of spleen, its direction, how it's placed in the body. You know about the surfaces. You know about the relations also. Now, important thing is we have to know, since we know it's wedge shaped, how many ends does it have, how many borders does it have. We know that it is having 2 surfaces, but we have to know about the ends. See, this is a little complex which you will have to understand. It has got 2 ends. This one is pointed, this is posterior end, and this one you will feel like it is a border, but no, this is a broad anterior end. Understand? So the anterior end is broad and posterior end is rounded, but compared to the anterior end, it is quite narrow. Clear? So this is the posterior end and this is the anterior end, which is so broad that it looks almost like a border, but it is anterior end. Now the borders. This is the superior border, this is inferior border, and this elevation you can see, this is intermediate border. Now you will say, what are the distinctions which will tell us which is superior, which is inferior? If we get the organ separately in our hand and we have to tell what is the posterior end, what is the anterior end, that's simple. The narrow end is posterior, the broad is anterior. The surface which has got impressions is the visceral surface. The surface which is convex is diaphragmatic surface. But the borders, one border, intermediate border, this is simple. The border passing through the hilum, where splenic artery, splenic vein, that means the vessels of the spleen, they are present. This border is intermediate. The only questions now remains is which is superior and which is inferior. So you just see carefully this border. This is absolutely clean and straightforward. No notch, no elevation, nothing. So the border which is smooth, clear cut, having no notch, no impressions, no elevations, it is the inferior border. And now just have a look on this. This is more convex and it is having notch here, see? So the border which is having notch at one end, here there is no notch, see? From this place to this posterior end, it is clean and smooth. So on one end, that means near the anterior end, it is having a notch. So the border which is having a notch at one end is the superior border, and the end near to which this notch is present is the anterior end. Clear? So this notch has not only told us which is the superior border, it has also told which end is anterior. Clear? So it has got superior border, inferior border, intermediate border, two ends, posterior end, anterior end. All the impressions are clear, the ends are clear. Now the blood supply, we have to talk about it. See, you can see such a clear and large artery. This is splenic artery. Splenic artery is the largest, in fact the largest branch of celiac trunk. Celiac trunk is the artery of foregut. Superior mesenteric artery is the artery of midgut. Inferior mesenteric artery is the artery of hindgut. Remember this. So the spleen is supplied by the splenic artery, which is a branch of and a large branch of celiac trunk. And by the time it reaches the spleen from the celiac trunk, it gives off a lot of branches. What are those branches? Pancreatic arteries, perpendicular pancreatic arteries, pancreatica magna artery. It gives short gastric arteries also. Means while coming, it is going to give short gastric arteries upwards, which supply the fundus of stomach. That means on the right side of the stomach, the fundus is supplied by the short gastric arteries given off by the splenic artery. And then the splenic artery reaches the hilum of spleen, where it divides and redivides into arterioles, which finally divide into capillaries, just like any other place in the body. And then the sinusoids are present. And sinusoids then collect together to form venules. Venules then join and form larger tributaries of splenic vein. And finally we have got splenic vein. It also comes out from the hilum of spleen, and then it reaches near the celiac trunk. where it joins superior mesenteric vein. But before it does that, it also receives the tributaries from the pancreatic veins as well as inferior mesenteric vein. But when it joins the superior mesenteric vein, both of them together behind the neck of pancreas form the portal vein. And portal vein is the vein which is having all the venous drainage from the abdominal organ, and all, and therefore it's containing digested food. So this digested food is sent to liver by the portal vein. Don't forget this. So this was the blood supply. But there are three theories about the type of blood supply. See, it's really surprising that nowadays when we see that we have got so many advanced technologies, still we are having various theories. We are not still sure what kind of blood supply it's really having. Some say it's an open vascular system, some say it's closed vascular system, and the third one is the compromised theory. So there are three theories: open vascular system theory, closed vascular system theory, and lastly the compromised theory. The compromised theory means the vascular system is both open and closed. So what is open and what is closed? Open and closed is nothing but when the capillaries, after the capillaries, the arterial system ends and only the sinusoids are present, which contain both WBC and RBC scattered. And later on these sinusoids, they join together to form the venules. This is the open theory, open vascular theory. The closed vascular theory says that the capillaries, they just don't open up just like that and spill their blood. Instead, they open in the walls of sinusoids, and sinusoids later on give the blood to the venules. So that's the closed theory. And compromised theory says that both are present. Means somewhere the sinusoid is open and sometimes the capillary is open in the walls of the sinusoids. And because of this reason, the interior of the spleen histologically is divided into white pulp and red pulp. It doesn't mean that it is white in color or red in color. The only thing is that in the periphery of the matrix of the spleen, we have arterioles surrounded by WBC. So WBCs are in full form called white blood corpuscles. That is why the periphery is called white pulp. It's not white in color, it has instead got a lot of white blood cells. So that is why it's called white pulp. Whereas in the sinusoids, since the RBCs are scattered, so it's called red pulp. So the center is called the red pulp and the periphery is called the white pulp. It is not because of white in color, as you may see. And you know that the spleen is lymphoid tissue. That means its function is, one is to prevent the infections. Second, in the fetal stage, it produces blood cells, so it's hemopoietic organ also in fetus. Then in adults, that means after birth, it also acts as a sieve. It removes the damaged red blood cells. So we cannot just remove all the RBCs because they are damaged, because every RBC contains hemoglobin, and heme means iron, globin means the protein, and we need both of them again to rebuild the hemoglobin, to again prepare a new RBC. So that is why here the damaged portion is removed and the iron and globin is taken up, especially the iron. So this is the function of spleen. Now, the applied anatomy. First of all is splenomegaly. In any condition, in any disease where there are a lot of malformed RBCs, except for sickle cell anemia, these malformed RBCs are going to be removed by the spleen. So the spleen will become enlarged. One of the very common reason of enlargement of spleen is chronic malaria in endemic areas where malaria is very common. So the splenomegaly means enlargement of the spleen. Another is rupture of spleen. Spleen may rupture also because of injury, any blunt injury or sharp injury, it can get ruptured. And since it is highly vascular, see how large the splenic artery is. So it is highly vascular, so it will cause a severe hemorrhage. And in that condition, we may have to even remove the spleen. Okay, so students, I think you have understood the spleen. It is a small topic but very important. So nothing is unimportant in our body or in this world, in fact, but read carefully, study carefully. It's very important but small topic and very easy topic. Thank you.